Welcome again to the May 2016 edition of the AJP-Heart and Circulatory Physiology video table of contents, or from the editor's desk. If you are in the Northern Hemisphere, I hope you're enjoying spring. If you're down under, I'm sure fall is in the air. Either way, you're going to love our content this month. If you peruse the journal, you'll see we've published 1 review, 3 rapid reports, 30 original articles, and 1 letter to the editor. I think we really have something for everyone. Here's a sampling.
Cardiac regeneration is a hot topic, but fraught with issues. In a review by Zembrowski et al., many of these aspects of understanding cardiac myocyte regeneration and proliferation in mammals are discussed. Understanding cell cycle factors and receptors may shed more light on getting this technique to result in significant regeneration. In one of our rapid reports by Borman et al., they examined the effects of aging on endothelial cell calcium signaling. The focus of this study was on the internal elastic lamina through which localized calcium signaling by endothelial projections occur. These investigators tested the hypothesis that local endothelial calcium signaling is impaired with advanced age, along with a reduction in the holes in the internal elastic lamina. In mesenteric arteries, the frequency of spontaneously local calcium signals in endothelial cells was reduced by 85% in old versus young animals. The internal elastic lamina holes were reduced by 40%. The author suggests that diminished signaling between endothelial cells and smooth muscle cells contributes to dysfunction of resistance arteries with advanced age. Lucar et al. Tested the hypothesis that inhibition of glucose-6-phosphate dehydrogenase prevents 20-heat synthesis, and that 20-heat-induced pro-inflammatory signaling promotes a secretory phenotype in vascular smooth muscle cells. They showed that G6PD inhibition decreased 20-heat synthesis in pulmonary arteries. This effect was also translated into reductions in 20-heat activation of mitochondrial superoxide and activated MAP kinase and phosphorylation of ELK1 expression. This is a novel regulatory mechanism whereby 20-heat can modulate various signaling pathways potentially regulated by glucose-6-phosphate dehydrogenase. Dale and co-workers investigated the influence of acute dynamic preload reduction on left ventricular strain and strain rate and the relation to preload recruitable work and positive DPDT. Speckle tracking strain and strain rate in the longitudinal, circumferential, and radial directions were measured during acute dynamic reductions of end-diastolic volume during 3 different myocardial inotropic states. Both strain and strain rate were sensitive to unloading of the left ventricle, but the load dependency for strain rate was modest compared to strain. These data suggest that speckle tracking echocardiography-derived strain rate is more robust to dynamic ventricular contractility. Finally, Alleman et al. Determined if preservation of mitochondrial membrane potential contributes to the antiarrhythmic effects of exercise. Hearts from sedentary and exercise-trained rats were perfused on a 2-photon microscopic stage for simultaneously measuring mitochondrial membrane potential and ECG. Exercise preserved mitochondrial potential following ischemia reperfusion. Mitochondria from exercise rats sustained respiration with lower rates of hydrogen peroxide production in an anoxia reoxygenation model. The authors conclude that exercise helps to sustain post-ischemic mitochondrial bioenergetics and preserved potentials, thus protecting against arrhythmia generation.
Thank you again for watching. One update on the journal. Next month we will begin posting articles as they're accepted, rather than you having to wait for a complete volume to be populated. Therefore, you will receive promotions for articles throughout the month. You can sign up for the e-talks of articles as they become available. And don't forget to listen to our latest and previous podcasts at the AJP-Heart and Circ webpage, where you can also see our current calls for papers. Don't forget to send us your best work now, and we'll see you next month.